Honored to introduce Fernando Schmitt, today's speaker on molecular cytology. Professor Fernando Schmitt is Professor of Pathology at the University of Porto, Senior Researcher and Head of Molecular Pathology Unit at the Institute of Molecular Pathology and Immunology at the University of Porto, the President of the Portuguese Society of Cytology, and also the General Secretary of the International Academy of Cytology. He has authored an impressive more than 435 papers in peer-reviewed journals, 23 book chapters, and is the editor of 3 books, one on molecular pathology and also in cytopathology and breast cytopathology. He's widely considered a world-leading expert in cytopathology and breast cancer. He was a fellow at the Karolinska Medical Hospital and was professor of pathology at the University of Sao Paulo, Brazil, and also at University of Toronto in Canada. His research work is on breast cancer with emphasis on molecular markers, cell adhesion and invasion, therapeutic targets, and mechanisms of resistance. In the last years, his research work has been dedicated to the study of molecular markers on cytologic material. He serves as the associate editor of 6 major scientific journals and sits on the editorial board of many others. He has also been chair, president, or scientific director of several major European, Portuguese, and Brazilian scientific societies of pathology and cytology. He received the prize of Educator of the Year in 2011 by the Papa Nicolau Society of Cytology and has received the Goldblatt Award in 2013 from the IAC. And with no further ado, I will turn it over to him. Thank you so much.
Thank you so much, Chris, for this nice introduction. Hello, everyone. It's a great pleasure and honor to be here to share with you our experience and what happened in the last 10 years concerning the application of molecular techniques on cytology. And I would like to start with this paper published in Acta Cytologica in 2007. And this paper, it was led by Dr. Marluce Bibo, that made a lot of questions for a panel of experts asking how technology is reshaping the practice of non-gynecological cytology. So in that time, we tried to do an exercise of prediction of the future and how we are in prediction of the future. So 10 years ago, we published this paper in Diagnostic Pathology called Gynecological Cytology, Too Old to be a Pop Star but Too Young to Die. And in this paper, we proposed that in the future, probably HPV test will be the first screening test for cervical cancer. And then we have reflex cytology in the positive cases. So in fact, this today is a reality in our country, as well as in Netherlands and Australia, and very soon in many, many other countries. So in fact, after the introduction of immunohistochemistry in the early 80s, we have a second revolution in pathology and also in cytology with the introduction of the genomic tests. And at the present moment, we are during the third revolution, that is the introduction of digital pathology that I will comment in the end of this lecture. If you look at the history of cytology, there are many ups and downs. For example, since the introduction, especially of fine needle aspiration in the Memorial Hospital in the early 30s, cytology is growing with the Papa Nicolau test and then the introduction of FNA and the spread of FNA from the Karolinska Hospital during the 80s. However, the introduction of the core needle biopsy and especially the lack of expertise of most of pathologists in cytology, there is a big down of the use of cytology in the 2000s. As you know, and we will comment later, lung cancer, unfortunately, is diagnosed most of the cases in advanced stages and should be managed only with the cytology diagnosis. Also, lung cancer today need molecular tests to guide the therapy. And this is produce a boost in the use of molecular techniques in cytology because we need to search, to study these biomarkers in the small quantity of material in these cases. And fortunately for all of us today, cytology is in the up phase. Because we know that it's worthwhile finding an actionable genetic alteration in lung cancer. As you can see here in this graphic, cases of lung cancer that has some genetic alteration, so that we can use a target therapy. When we use this target therapy in these patients, compare with the classical treatments like chemotherapy, these patients goes well and have more survival. This diagram was published by our group in 2011, showing that in lung cancer, it's important to do the diagnosis and to distinguish two big groups, the small cell and the non-small cell. However, we know that's not enough just to say this. We need to distinguish the squamous cell carcinoma from the adenocarcinoma, because the adenocarcinoma has a lot of genetic alterations that are targetable in our days. So all these starts with the EGFR, and if the case is EGFR positive, the patient can be treated with tyrosine kinase inhibitors, but it was not positive in the second test we do ALK, and if it is positive, this patient is eligible for crizotinib, but many other targets in the last years appears in lung cancer, showing to us that it's not possible to do one test after other tests. We need to find them, and in fact, the labs found the technology that can study a lot of genes and few quantity of material. So this changed the paradigm of the pathology and cytopathology, because small biopsies and cytological samples should be managed not only for diagnosis, but also to maximize the amount of tissue available for molecular studies. And if you compare these two tables in this paper from Salto-Telles, we can see that after 2004, more and more molecular tests is being applied in cytological material, increasing the importance of cytology, not only in the diagnosis, but also for the prognosis and the indication of therapeutics in the patients. So after this paper published in 2007, what happened 10 years later? So during the anniversary, 60th anniversary of Acta Cytologica, we do some important papers published, and we try to look back and to see what happened. And then in 2017, we published this paper about what happened 10 years after these expectations and projections for the use of ancillary techniques in cytology. So one of the questions raised 10 years ago is that the role of morphology. So Dr. Bibo asked to the panelists, do you think morphological features will still play an important role 10 years from now? In that time, all participants predicted that morphology would still play an important role 10 years later. And in 2018 now, and certainly in the next years, morphology still will be the cornerstone for the diagnosis in pathology and cytopathology. And we know more and more that the success of molecular techniques still depends on the good qualitative and quantitative evaluation of the cytological material. So the value of traditional morphology has not diminished. It is simply no longer sufficient. So we need today something more than the morphology. For example, to apply morphological or ancillary techniques in cytology, morphological control is one of the most crucial issues in the application of these techniques. And the role of cytopathologists is mandatory in the collection and selection of cells. So this is the key points to use any molecular techniques in cytology. Collect good and well-preserved material, validate the molecular studies on cytological material, and specifically control the cases morphologically. This consensus conference sponsored by the European Society of Medical Oncology for pathology and molecular biomarkers for lung cancer specifically say something that is very important for us pathologists. Of course, they say that we can use classical biopsies, we can use cytological samples, but this is important. The same pathology should, if possible, review all available tumor material from the same patients, including biopsies and cytology specimens. Also very important for all of us, a pathologist should be involved in the sample preparation for DNA extraction. So these are the different steps for the molecular testing. So we can start with the samples. We have a pre-analytical phase where our job is very important in to select the areas, in to collect the material well, in to fix this material well. We have an analytical phase in the laboratory that starts with the DNA extraction and go to the platform, go to the interpretation, and also pathologists should be involved in this analytical phase, especially in the interpretation, in to put in the context all the results produced by these different technologies. So this is an example in our material, in our laboratory. Always the pathologist involved, the pathologist select, in this case, it's a core biopsy, but in cytology is the same. We select the area, we give the amount of tumor cells, and all these informations are important for the conclusion of the molecular test. Why? First, because the percentage of the tumor cells are related with the detection sensitivity of the methodology. For example, for Sanger sequencing, the method sensitivity is set up at 20% of tumor cells. So if we have less than 20%, we put a comment, especially if the test is negative. Look, the test was negative, but I have less than the minimal established in my lab to do this test. For example, this is a case that we received in consultation. We received first the biopsy, and as you can see here, there's only these groups of malignant cells. And we perform molecular, but we state that there are less than 20% of tumor cells, and we didn't find any mutation. So, as our laboratory is a reference laboratory, and we receive this biopsy from another hospital, I call the pathologist and say, look, you have more material from this patient? And the pathologist said, okay, I have cytology, but I didn't know that cytology is a material to do molecular tests. And this is one important thing, because we need also to teach or to inform our colleagues pathologists that cytological material is also, and it is good if it is not better than histological material to do molecular tests. So there are smears from this patient with a lot of tumor cells, as you can see here. We scrape the smear, and in this case, we found a mutation in exon 21 of the EGFR. So we decided to do an analysis in our laboratory about the quantity of tumor cells and the frequency of detection of mutation. This is for all kinds of tumors, all different mutations, but it is very impressive that when we have more than 20% of tumor cells, we detect almost the double of mutations when we have less than 20% of tumor cells. Okay, it's important to know which molecular test we will use, because the amount of tumor cells depends on the information that you want to provide to the clinicians. For example, if I'm interested in detect hotspot mutations or fusion drivers, I need 20% of tumor cells in my sample. However, if I am interested in to inform also about copy number variants, we need to have 50% of tumor cells. So we need to be informed about this, we need to know about this to do the correct information and to interpret in the end the results that we obtain. We need to select tumor cells in general mixed with the benign cells. So today, artificial intelligence is playing a role in this selection. Automatically, especially, and this is what's tested for histological sections, it's possible to detect the tumor cells. The machine, you can digitalize the slide, and using software, you can detect the tumor cells, and the machine did the absolutely calculation how is the percentage of the tumor. And this has a very nice correlation with the visual estimation made by the pathologist. So, since we mark the area in the slide, when the slide arrived in laboratory, the technicians are able to do macro dissection from this area, as you can see, and then they scrape the material for an Eppendorf tube, and then extract DNA and do the analysis. Sometimes we have a very cellular cytology, but with a low tumor fraction. And we need to interpret this, and we need to give the exactly, or not exactly, but approximately quantity of the tumor cells, for example, in the middle of inflammatory cells. We can use, in some specific cases, laser microdissection. For example, when we have low tumor fraction, we can isolate, we can mark, and we can isolate only the tumor cells in the Eppendorf tube for the molecular analysis. So, the second question in that paper is about the role of the imaging techniques. And for example, one question is that can the imaging techniques replace cytology in diagnosis? Although there was many advances in radiology in the last 10 years, until now, no imaging technique replaced morphological diagnosis. What about the role of immunocytochemistry? So despite collaborative studies, improvements in the technique, automation, better quality control, we still do not have an universal standardization for the application of immunocytochemistry in cytology. So, because especially we have different fixations, we have different types of preparations. And the best preparations in this moment in our hands is cell blocks. So, most of the diagnostic and prognostic tests using immunohistochemistry 10 years ago are still in use. However, one of the markers that is extremely useful today for the subtyping of non-small cell lung cancer, the P40, appeared in these last 10 years. Interestingly, none of the panelists predict the screening role of immunocytochemistry for molecular tests, as ALK and ROS1 antibodies used to select cases for searching translocation in these genes. So, in 2011, we did this study trying to find why the results of the immunocytochemistry are so variable from the different laboratories. And the variability in the type of material and in fixatives was the major factor that prevented standardization in immunocytochemistry. We conclude, for example, that the methods of fixation and the antigen retrieval are key points in the staining process for immunocytochemistry. So, we couldn't demonstrate in this study that formalin fixation was better than alcohol or methanol, etc. We clearly demonstrate that when the antigen needs retrieval, we need to do antigen retrieval in cytological material, and this works very well. So, what about the P40? So, P40 is a very important marker to demonstrate squamous cell differentiation in non-small cell lung cancer. So, basically, we found in this meta-analysis that P40 is 97% specific for squamous differentiation, in contrast with the P63 that is 69. So we agree with some colleagues that say no P40, no squamous in lung cancer. So once we diagnose the subtype of non-small cell lung cancer, So we pass for the molecular tests, and we can use immunohistochemistry to screen some molecular alterations. And one example is for ALK. So we can do immunohistochemistry, for example, using this clone B5F3, and there is an extremely high correlation between positivity and presence of translocation, and especially negativity and absence of translocation. So we can use these clones and also the 5A4 clone with high accuracy in immunocytochemistry. And we can put in this algorithm. So if the ALK by immunohistochemistry is negative, there is no rearrangement. And if it is positive, we can confirm the rearrangement using FISH. The same algorithm can be used for ROS1, other important molecular alterations in lung cancer. So we can use exactly the same immunocytochemistry. If it was negative, we can avoid to the FISH. If it is positive, we can confirm by the FISH test. Another use of immunocytochemistry that is coming in lung cancer is the immuno markers for immuno checkpoints, the markers for immuno checkpoints. Because the introduction of pembrolizumab, anti-PD-1 drug, showing a very nice improvement of survival when compared, for example, to the chemotherapy in patients that are positive for PD-L1. So the pathway PD-1, PD-L1 in general can be abrogate by the tumor cells that express PD-L1 and suppress the activity of the T cell. So if the tumor cell express PD-L1, we can use drugs that inhibit this interaction. And the inhibition of this interaction restores the T cell function, and the T cell can attack and destroy the tumor cell. So PD-L1 can be detected by immunohistochemistry in tumor cells and immunocells and in lung in normal alveolar macrophages. Of course, that it's important the assessments in tumor cells for some drugs and in immunocells for other drugs. How to choose the PD-L1 antibody is an important discussion in lung cancer. Why? Because we have at least 5 different drugs with different antibodies and with different cutoffs. And because each PD-L1 inhibitor has its own assay, and clinical trials have assessed different levels of PD-L1 expression for correlation with clinical outcomes, there still is confusion about the use of PD-L1 levels. And the common questions are the results between different assays interchangeable? What level of expression is clinically relevant? Is an actual biopsy specimen required, or can an archive sample be used? Is all diagnostic material suitable for PD-L1 testing? Are there specific requirements for the laboratory performing the test? So there are biological problems because the expression of PD-L1 in lung cancer could be heterogeneous, as you can see here. And of course, if the sample is from this area, the test will be positive. If it is from this another area, the test will be negative. There are very few studies using cytological samples for PD-L1. This is one of the studies show an overall concordance when you use the cutoff of more than 1%, 85%, and 94% of concordance when you use the cutoff of more than 50% of positive cells. Due to these many issues about the expression of PD-L1, so expression can be missed in the small specimens, for example, in cytology. The expression is dynamic and can vary with the time. The PD-L1 epitopes are potentially unstable and can vary with the fixation. So there are so many issues that as we can say that PD-L1 needs a psychiatrist to sort them through these issues. So we need other candidates to study the presence of alterations that show this tumor can be targeted by immunotherapy. And there are all potential candidates. The most commonly studied at this moment is the tumor mutational burden and the microsatellite instability. We know that the more immunogenic tumors, that in general are the tumors induced by carcinogens, like lung carcinoma, melanomas, gastric carcinomas, colon carcinomas, these tumors more frequently respond to the immune checkpoints drugs. For example, this other paper recently published in the New England Journal of Medicine showed that the use of nivolumab and ipilimumab in lung cancer cases with high mutational tumor burden, they have better survival when compared with the use of chemotherapy. So maybe one alternative in the use of PD-L1 in cytology is to use next generation sequencing to assess the tumor mutational burden in these cases. So back to our paper. The other question is about the role of clinical cytogenetics in the use of in situ hybridization. So although some authors predict the expansion of the use of FISH, in fact, in this last decade, the use of FISH in cytology is practically the same. The indications for using the Eurovision kit in urinary cytology is still under debate, as well as its role in the diagnosis of effusions and pancreato-biliary cytology. In fact, there are recent papers showing that NGS can replace the use of FISH, for example, in the diagnosis of effusions and pancreato-biliary cytology. Like 10 years ago, FISH is used for detection of translocation and sarcomas and lymphomas, but it is limited to some specialized centers. Interestingly, one of the most common use of FISH in cytology in the last years, the search for out translocation in lung cancer was also not predicted in that time. So this is an example of the use of multiprobe FISH, the Eurovision, that looks the ploidy status for chromosome 3, 7, 17, and the deletion of 9p21 locus. This can be applied also to pancreato-biliary cytology and also for the detection of 9p21 deletion in effusions with malignant mesothelioma. There are other examples for the applying of the FISH in soft tissue tumors. Basically, the use in cytology is limited to confirm recurrences because in the primary tumor, it was preferable to do core needle and to study these cases on histology. So, as I told you, we can use FISH to detect out translocation and non-small cell lung cancer patients. This occurs in 1 to 70%. In general, the patients are younger, no history of smoking, and they are adenocarcinomas. So here you can see an ML4R translocation in lung cancer. We have a split apart of the two probes, green and red, here, and here. So this is a positive case. So what about the role of the molecular techniques? 10 years ago, the use of molecular techniques to detect EGFR in lung, KRAS in colon cancer, and KIT and GIST was just started. Today, as I mentioned before, in almost 50% of lung cancers, the only material available for molecular studies is cytology. These change the paradigm because we need to maximize this small material for use in molecular application. Again, a technique that is revolutionizing cancer genomics, the next generation sequencing, was not even mentioned at that time. And this technology is rapidly replacing the classical use of PCR and Sanger sequencing allowing to study not only panel of genes, but also RNA alterations. 10 years ago, the idea that all of the genes altered in cancer could be identified base pair resolution would have seemed like science fiction. But today, such genome-wide analysis through sequencing of the exome or even the whole genome is routine in many pathology labs. And why now these techniques are being used more and more? So first, because of the Human Genome Project. We know our genome. Second, the Cancer Genome Atlas that did a map of 33 different tumor types, including the most commonly and 10 rare cancers. So we know the genome anatomy of the tumors. And third, the presence of molecular tools. Today, we have much more potent molecular tools that enable us, in a reasonable price, study a lot of different alterations on DNA and RNA. So we saw this evolution. We start with single markers that study 1 to 5 genes. For example, we can see in lung that starts with EGFR. So we are in the phase of cancer panel. So with the diagnostic companies, so 10, 300 genes. And probably we will move for the whole exome, studying around 21,000 genes. So let's back to the lung cancer, because this is the best example of the use of these techniques in cytology. As I mentioned, from EGFR and all, many other drivers were detected in lung adenocarcinomas. As you can see here, ROS1, BRAF, HER2, RET, MET, more recently, NTRK1 fusion, etc. And the ideal test to cover the most common altered genes in lung cancer is a multi-test single assay like next generation sequencing, that instead to use and to study just one fragment of DNA, are able to study and to cut and to study different areas, different genes, all in one shot, all in the same time. However, so only DNA, it's not enough, as these authors show, that if we study more and more genes, and together with these genes, we study the transcriptome of the tumor, we are able to cover most of the genomic alterations of the tumor. For example, for lung cancer, in our laboratory, we use this commercial panel at this moment that cover the most frequently altered genes that can be targeted for the therapy. Probably, next generation sequencing will replace completely the single gene approach. So according to this prediction for the National Institute of Cancer in France, published in 2015, until 2019, all the single gene approach will be replaced by target NGS, and maybe in 2020, half of the studies will be done in the whole exome sequence. What I can say to you that in our laboratory, this phase already happened. So we are not using lung cancer more single gene approach. We replace completely this approach by target next generation. The molecular tests in our laboratory are increasing year after year, as you can see here. And these graphs show very well that the detection in 2015 of EGFR and ALK at the same time when compared with 2017. So this is EGFR and ALK practically disappeared and what replaced it by next generation sequencing. Can we use next generation sequencing in cytology? We can use, these are the two most common commercial platforms available for next generation sequencing. All of these commercial platforms accept cytology as a sample, a kind of material that we can use to do sequencing panels. So in our laboratory, in 2013, we validate the use of cytology for next generation sequencing. So with just 10 nanograms of DNA, we are able, for example, to run panels looking for hundreds of mutations in different genes and tumor suppressor genes. So we can use smears, we can use cell blocks, and we can also use liquid-based cytology. And the smears can also be previously stained by Papa Nicolau or liquid stains. In the recently guidelines published for the molecular testing in lung cancer patients, this statement appears that pathologists may use either cell blocks or other cytological preparations as suitable specimens for lung cancer biomarker molecular testing. So we can use smears in a very confident way, and this is in the guidelines and in nowadays to study biomarkers in lung cancer. Of course, that there are advantages and disadvantages in use different preparations in next generation sequencing. For example, direct smears is very good, but we need to sacrifice the slide from the archive. So in this case, the use of digital pathology is useful because we digitalize the slide that we will use, then we keep the slide in the image in the slide, and we can destroy to do NGS. LBC is also a good methodology, but need additional validation because of the fixation uses. And cell blocks that are very easy to validate because most of the laboratories of molecular pathology use paraffin blocks, and the technique for use cell blocks is practically the same. We need to be conscious that there are several pre-analytical factors that can affect the NGS analysis cytology, like the specimen cellularity, the type of preparation, the type of fixative and stains, the type of glass slides used, the tumor fraction, the DNA yield, and the input DNA for the study. We need to be prepared, and if in your hospital, in your clinic, it is possible to do rows, for example, in the collection of material in lung cancer, this will be very, very important, because during the rows it's important not only to assess, okay, material is there, but also to assess the quantity of material that is there, and if you need additional passes, and if you need to keep more material for molecular tests or other tests depending of the pathology that we detect. Always that we put cytology in the equation, many concerns came, for example, but cytology is reproducible for the molecular tests. There are consistency in the use of cytology for the molecular tests. For this reason, this study was done. So a ring trial study on quantitative cytological molecular reference that were done by 16 different laboratories around Europe and North America to assess if the material, cytological material with no mutation in the smears, if these mutations in different allelic fractions can be detected by different platforms, different routines used in different laboratories. And we found in this study a very high reproducibility in the detection of the mutations with a very good performance of NGS, regardless the platform or the gene panel used in the different laboratories. So the answer is yes, cytology is consistent and reproducible to apply next generation sequencing. Other tests are coming to the pathology molecular lab. For example, the IDELA tests that use this PCR test for detecting 2 hours mutations in KRAS, mutations in BRAF that can be very, very useful, for example, in melanomas in fine illustration of metastatic melanomas or effusions, we can apply this test very easily. And recently, it was demonstrated that this test is also consistent with the use of cytological. So, we are going to the end and we insisting in lung cancer. Because in lung cancer, there are more than 300 non-synonymous mutations and that are established targets like EGFR, ALK, ROS1, BRAF, and PD-L1. So the panel that we use at this moment to screening alterations in lung cancer include the most common genes like EGFR, KRAS, BRAF, also include the fusion drivers such as ALK and ROS1. We know that in lung cancer, another important use of cytology in that lung cancers that progress under the therapy. For example, if you have a tumor, and this tumor was detected an EGFR mutation, sensitive, for example, to gefitinib and erlotinib. If during this therapy the cancers progress, one of the reasons is because a small clone grow, and this small clone show a secondary EGFR mutation. Most common is T709M mutation. This is a resistant mutation. So, if this mutation was detected, now it's important because it's more important because there are specific inhibitors for this mutation. And then we can treat this patient with these other drugs and the survival is significantly improves. So, but in a patient with an advanced lung cancer that progress, could be difficult to do a new biopsy of this patient. So one alternative is to detect these alterations in circulating tumor cells or in circulating free tumor DNA. However, if this test was negative because the sensitivity is not so high, we need to study this mutation in the tissue. And cytology is a very good approach. For example, imagine if this patient has an effusion, we can drain the liquid, and if there is tumor cells there, we can apply the test there. Or we can aspirate some metastatic site on the liver, or even in lung or lymph nodes, and to study this cytological material. Because cytological samples are suitable for testing for this mutation using highly sensitive and qualified method like NGS. Let's go to the conclusion. If you look at the conclusions for this paper in 2007, we can see that one of the most important discussions still deserve reflections, because they say it's important that we cytologists take an active role in the adoption and application of molecular techniques, since we are able to interpret the results in the light of cytomorphology. If we do not act, others will take over. This is in 2007. The advance of molecular techniques poses two challenges for the cytology laboratory: deciding when to adopt a new test and deciding who should perform and interpret. The cytology laboratory will likely find itself squeezed between rising expectations for advances in molecular medicine and limitations imposed by good evidence-based practice and funding. All these sentences that I took from the original article in 2007 are still very real and very present, because we will still are dealing with these problems. So, 10 years on, cytopathologists and pathologists in general still do not play an active role in the adoption and application of molecular techniques. There is no doubt that many things change, and there are more pathologists involved in molecular pathology. For example, molecular tumor boards start to be a reality in many oncological hospitals. However, there is a gap between the fast implementation of the technology and the slow engagement of the pathologists. In some laboratories, it's possible to see pathologists working actively together with molecular biologists and scientists, deciding with them the implementation of new tests and sign out of the reports. Funding is still a problem, but many tests used are tailored to specific treatments. And in many cases, it's possible to demonstrate the financial advantages to implement the tests. What's coming next? The study of fluids, blood, so liquid biopsy is in reality, the detection of mutations in the urine for solid visual cancers, the use of the cerebrospinal fluid, the use of epigenetics, whole genomic sequencing, as we mentioned before. Of course, there are many challenges, but in reality, this is happening more and more. Cytopathology is changing. We can use cytopathology for many other applications. For example, breast tumors, small tumors now are operated. So how to banking this tissue? So we need to use sometimes all the tissue for the pathology diagnosis. Alternative, aspirate the fresh tissue and then banking the cells instead of banking the tissue. Today, PDX is a reality in some hospitals. So we can aspirate, for example, tumors. We can xenograph in rats and we can treat these rats and to see how these how they respond to the therapy. More and more, as I show in this presentation, cytology can be used to sample metastases because we know that metastases are different from the primary tumor because we know very well the tumor clonal evolution and we know that the tumors are heterogeneous itself and the tumors are heterogeneous in the different metastatic sites. So after 10 years, we can conclude exactly as Dr. Bibo concluded the original discussion. Since molecular biology results are meaningful only when interpreted in the proper morphological correlation, it is important to standardize molecular techniques and organize education in molecular biology. So we are in this phase because we are trying to converge classical, molecular, pathology and cytology for one diagnosis. But we need to be very, very attempt because this can be again do these ways. For example, in 2007, the first tissue agnostic drug was approved. So pembrolizumab received approval for any tumor, independently of the histology, that have microsatellite instability. More and more, we are seeing basket trials that are based on the same mutation in the different histological tumors. More and more, we are seeing tests like this one. So with the genomic alterations identified and with the therapeutic implications for these different genomic alterations find and identified in the tumor. So very complex tests with a lot of new data and correlating results with therapeutic response. But we need to keep our proud as pathologists. We believe that molecular pathologists belongs to the pathology. So in different organs, the application of these techniques are essential, but they are guided by the morphological study. We need more and more to participate in multidisciplinary discussions together with our colleagues, our surgeons, oncologists, immunologists, genomic medicine experts. So our role there is absolutely essential, and this is still essential in our days. And digital pathology is coming to integrate all this data. So this will be, or it is in many cases, the tools in the classical pathology laboratory this moment. Not more only the classical instruments that were used to have, but also instruments for molecular pathology. And these can be integrated in the digital pathology. In the digital pathology with one final conclusion with all the information that we receive. So, small samples like cytology, exploring morphological and molecular information are the model biopsies of the future. Several techniques using different types of cytology have shown good results with similar or higher accuracy when compared with surgical specimens. And presently, with the rapid development of personalized treatment, each tumor should be tested for available biomarkers, and every effort should be made to spare the tissue for molecular testing. Thank you very much for your attention, and I'm happy to answer any questions. Thank you.
Thanks. That was an amazing talk. I really like the organization going from the article 10 years ago and breaking it down. It's very comprehensive. Appreciate it. I just wanted to tell the participants, if you have questions, you can use the chat box to type the questions in, and then I will address the question to the speaker. So I'll let you have a few minutes to locate the chat box and type your questions in. In the meantime, I just had curious about testing from smears because we're not really set up to do that much at my institution currently, but obviously this can be an important thing to do, and cytologic material provides good quality DNA typically. I noticed like you had an arrow coming from the diff quick stained smear, and I'm just curious if it's something that can be performed on using PAP stained smears as well.
Yes, Chris, thank you for your question. We didn't have any significant difference in the quality of the DNA extracted from DiffQuick previously stained slides and Papa Nicolau previously stained slides. So both stainings works for the most common molecular tests that we perform, but they works in a very similar way.
Thanks. Are you able to use smears that have been stained with the immunocytochemistry?
Yes, this is also very good questions, because sometimes we received some smears previously stained by Papa Nicolau, for example, with the presence of tumor cells are difficult to detect, to isolate the cells. So in some situations, are not common, but in some situations, we stain with immuno by TTF1, for example. So we detect the neoplastic cell by TTF1, and then we extract the DNA from these cells previously stained by immunocytochemistry. This is one example. Other examples, there are sometimes the only material available with cells are, for example, smears that the original pathologist stained by immuno, but we can use, and the results are still good.
That's very interesting. But just to clarify, in one of your examples, did you say that you had a PAP stained smear that you then destained and stained with immunocytochemistry, or you just did both on the same smear and then?
We did. When we perform, for example, immunocytochemistry in previous Papa Nicolau stained slides, we don't destain it, because during the immunocytochemistry in general they lose the original staining, and in the end we do counter staining with the hematoxylin. So we just remove the cover slip and we do immune.
Thank you. There's two questions queued up. The first one is it a good practice if there's enough material to perform a PCR test first and after NGS for other alterations?
So depend on the platform that you have in your lab. For example, if you have available NGS and of course the difference of, because today the difference of price are not so high, you know? But if you have the two methodologies available in your lab, we prefer to use NGS first.
The second is a comment. It says, thank you very much for a nice lecture and new scenery of the future.
Okay, thank you.
So I'll just wait another half minute in case there are follow-up questions or additional questions. I did have an additional question. Do you ever, if you have to, do you ever have to sacrifice a smear for testing that may be the only or most of the diagnostic material, and in which case do you image the slide?
Yeah, this has happened. It's not rare to happen. This happens with some frequency. So in this case, we digitalize the slide first, and then we sacrifice. If you have a lot of material, sometimes we can use half of the slide, although we always digitalize, but we can use half of the slide, we scrape and we preserve the other half. But if it is not possible to do, we just digitalize and then we sacrifice the slide.
That's brilliant. Yeah, I think this was a great session. For those of you that came in a little late or you want to review the lecture, it should be by tomorrow posted through the IAC link of webinars. And so you should be able to view the recording in conjunction with the slides. So again, thank you very much, Dr. Schmitt. It was great. I hope to have you back sometime. And thank you everyone for participating, and just keep an eye on the schedule. We should have another speaker in another month or two. So we have a few lined up throughout the year. So thanks, and have a great rest of your day.
Thank you very much, everybody. Bye-bye.